Good evening, everyone. So today we are going to look at the physiology of thirsty. So before we can dive deep into the concept of thirsty, let's first try to look at one of the concepts or the terminologies that we need to know so that we understand nicely. So under the physiology of thirsty, the concept that I would like to introduce you to, the first one, we have osmolality. Then we also have osmolarity. So basically these two concepts, they talk about the same thing, but they have got a difference that makes them to be different, something that makes them to be unique from each other, but they have one thing that they usually concentrate on or that they talk about. So when you talk about osmolality, which is this one with an L, this is basically just the concentration of solutes, the concentration of solute particles per kg of a solvent. So meaning that you are looking at how that particular fluid compartment is concentrated, how much solutes are concentrated in that particular body fluid compartment, of which that fluid compartment is being measured per kg. So osmolarity is basically the concentration of solute particles per kg of a solvent, and mostly, or rather let me just say, osmolarity is measured in milliosmoles per kg. So it measures the concentration of solute particles per kg, and its units is milliosmoles per kg. Yeah. So this is what osmolality means. Then when you talk of osmolarity basically also is just the concentration of particles. Now this concentration of particles, they are measured per liter, not per kg. So this one is just the concentration of particles per liter of a solution or in that particular solvent. So this one, it is per liter. So meaning that the units that are for osmolarity, it is milliosmoles per liter and not per kg. So they both talk about the measurement of the concentration of solute particles, but the difference is that osmolarity, it just talks about the measurements of those solute particles per kg of a solvent, but while osmolarity, it is per liter of a solution. So the one that is preferred between the two is osmolality. That is because osmolarity, it is independent, or rather let me say it is temperature independent, and it gives more accurate when it comes to the measurement of the solute particles. That's why osmolarity, it is the one that is preferred. So osmolarity, it is the preferred one because it is more accurate and it is temperature independent. Then the other concept that we need to be familiar with is it is tonicity. Tonicity. So basically when we talk about tonicity, it is just like we are describing the ability of a solution to cause water movement across a semi-permeable membrane. So tonicity, basically, it is a way of expressing the effectiveness of the osmolarity of a solution. So what you are going to know under tonicity is that it is just going to be there to express the effectiveness of osmolarity in a solution. So tonicity has got three types of solutions that you can look at, or three types of fluid that falls under tonicity. We have isotonic solutions, which we know isotonic, which we are basically aware of, and these terms are quite familiar. And we also have hypotonic solutions and also hypertonic solution. So you are going to find that in these three solutions that we are having, the osmolarity is not going to be the same. So meaning that if a cell is put in any of these three solutions, its reaction or the outcome of that particular cell will be different. That is because the concentration of the solutes in this particular solution is different. That's why we are saying that tonicity, it is just the ability of a solution to cause water movement across the semi-permeable membrane, because we know that in an isotonic tonic solution, you're going to find that the concentration of the solutes outside as well as inside of the cell, it is equivalent. But in an hypotonic solution, you're going to find that the concentration of solutes that is going to be on the outside of the cell and inside of the cell is not going to be equal. Same applies to hypertonic solution. So you're going to find that the outcome of a cell in these three solutions is going to be different because the osmolality that is in these cells or in these solution types of solution is different. So tonicity is just a way of expressing the effectiveness of osmolarity, and we know that that osmolarity, which is the concentration of those solute particles in that fluid, will create an osmotic gradient that will allow the water molecules to start moving across the semi-permeable membrane. So these concepts are very, very important as we are also trying to understand the body homeostasis, body fluid compartments, as well as the physiology of thirsty. So having looked at this, let's now try to also look at another term that is known as water steady state. Water steady state. Yeah. So basically, water steady state, it is just the balance between the water that we take in as well as the water that we lose. So we know that in our daily life as we live, no one can live days and weeks and years and months without drinking water or without taking water in any form of food. So you're going to find that the water that we take in, maybe in form of drinks that we take, as well as the food that we take, because we know that, for example, in Shima, you cannot make in Shima without water. That's kind of impossible. So you're going to find that the water that we take in through the drinks that we take, as well as the food, has to be equivalent to the water that has to come out. So the water that comes out, for example, the way we tend to lose water through sweating, if you are exercising, or maybe the time it is getting hot, like right now we are experiencing the hotness of this place, so find that you'll be sweating and you'll be feeling thirsty, so because you are losing water, as well as the water that we lose through the fecal matter, which is the feces, the urine, and also respiration. So you're going to find that the water that you are taking in has to be equivalent to the water that is coming out. So water steady state basically just talks about the balance between the water that we take in and the water that we take out, of which the water we take in is equivalent to the water that has to come out. So for example, if you are taking in 2 liters of water per day, it means that you also the amount of water that you have to lose through the feces, the sweating, the urine, the respiration, and so on and so forth, it has to also be equivalent to 2. Yeah, so you are going to find that the maintenance of the body water is quite an important thing for body homeostasis, because it helps in the functioning of the cells. Just like the way we talked about the tonicity. You're going to find that if you lose more water and you allow the concentration of the solutes to increase, it means that you're increasing the body osmolality. If the body osmolality increases, you're going to create an osmotic gradient that will allow water molecules to start moving from inside of the cell going to the outside. So the cell will shrink. So meaning that even the functioning of the cell is going to be affected because of its environment. So in order to compensate for that, the body has to come up with an homeostatic mechanism that to maintain that, of which we are going to look at one of it, which is thirsty. So now let's now talk about what is thirsty. So having known this concept, let's talk about now thirsty. So you are free to ask questions. Yeah, where you are not clear, just type. I'll be able to see. So thirsty. So thirsty is just the conscious desire to drink water. Just that conscious desire that just comes, say, hey bro, go and drink water. That is what we call thirsty. That is because the body has already detected to say the amount of water that you're having in you right now, it is not the required amount of water to support the body homeostasis. So it will create a mechanism that will give you a conscious awareness to say, drink water. So it is also part of body homeostasis, as we were talking about. So that's why homeostasis is very, very important. You need to understand this. If you haven't checked out our video, please go and check it out, then you'll be able to grasp the concept here. So now let's talk about the mechanism that is involved in thirsty. So the mechanism basically just going to help us in terms of knowing how does the body send signals and what mechanisms or how does it compensate for the same loss of water so that one can go and drink and Okay, so when you talk about the mechanism of thirst, one thing that I would like to help you know is that there are what we call osmoreceptors. So these osmoreceptors, these receptors are found in the hypothalamus. So we have the hypothalamus. So let's just try to imagine that this is the hypothalamus that we're having. So here in the hypothalamus we have got nuclei that are found here. There are a lot of nuclei. We have got the nuclei for thirsty, for the temperature regulation, and so on and so forth. So in the hypothalamus we have nuclei that are known as the organum vasculosa of lamina terminalis. It is abbreviated as the organum vasculosa of lamina terminalis, as well as the subphonic organ. So these nuclei, they have got receptors that are known as the osmoreceptors. These osmoreceptors are found outside the blood-brain barrier. So the brain has got what we call the blood-brain barrier. This barrier actually it is the one that regulates what actually goes in and out. We know that a barrier, like a membrane, it is a barrier. So it regulates what goes in and out. So you're going to find that these the hypothalamus, specifically at that region where we have these nuclei. These nuclei have got osmoreceptors. So these osmoreceptors are found outside the blood-brain barrier. So they have got direct contact to the blood plasma, such that they are able to detect the amount of solutes that are in that blood plasma, as well as the amount of water. So what happens is that a stimuli can come. So that stimuli can be due to you are sweating and you are losing more water, or you are having more solute gain, or you are having water depletion that is in your body, maybe due to vomiting, or you are having diarrhea. So in that condition you'll be losing water. So what will happen is that these receptors, let's assume that these are the receptors, these receptors are going to detect the change in the blood plasma. So when they detect that change, for example, you are having the loss of water due to sweating, you're going to find that when you lose more water, the concentration of the solute is going to increase, meaning that you are increasing the plasma osmolality. This is same applies to say you are having a cup of water and the water is here. Then imagine at this level, that's where you are putting in, that's the level of the solutes. Then you start losing water in this cup. So you're going to find that the level of the water is going to start reducing, and when the level of the water reduces, the concentration of this solute is going to increase. So you are going to have high osmolality. So when this happens into the blood plasma, when the osmolality is increased, these osmoreceptors are going to detect. These osmoreceptors are just basically the nerve cells. So when they detect, what will start happening is that they start losing fluid. So due to that loss of the fluid, they'll start shrinking. So the shrinking in those nerve cells is what will be sent as a signal to the organ nuclei that are found into the into the hypothalamus specifically. So they are going to be sent there to this nuclei, which is the organum vasculosa of lamina terminalis, as well as the subphonic organ. When they are sent there, what will happen is that a signal is going to be sent from now these organs. It will go now to the posterior pituitary gland, and when it goes to the posterior pituitary gland, it will initiate the release of a hormone that is called the antidiuretic hormone, which is abbreviated as ADH. This antidiuretic hormone, when it is released, it is going to increase water reabsorption in the kidneys. That is because the antidiuretic hormone has got what we call antidiuretic hormone receptors that are found in the kidney. So when it goes there and it binds there, it increases water reabsorptions in the kidney, and that mechanism will compensate, will try by all means to compensate for the water that has been lost. But that is not actually enough. So what the body does, or what happens also, is that these osmoreceptors, they are also going to activate the thirst centers that are found in the hypothalamus. As these thirst centers are activated, they are going now to trigger a conscious awareness that you have to drink water. They are the one that are going to bring about that conscious awareness to say you have to drink water, and then you're going to drink to compensate for the water that has been lost. So basically, just to summarize the mechanism, we know that increase in blood plasma osmolality, that can be due to sweating or solute gain or water deprivation in the body. It is going to cause a change in the plasma osmolarity, which is, it is going to increase the plasma osmolarity. So as the plasma osmolarity is increased, it is the increase is going to be detected by the osmoreceptors that are found in the hypothalamus. So when the osmoreceptor take that signal into the nuclei that are called the nuclei that are known as the organum vasculosum of lamina terminalis, as well as the subphonical organ. So as that happens, these then are going to send signals to the posterior pituitary gland, which will bring about the release of antidiuretic hormone, which will cause water reabsorption in the kidneys. Then they are also going to activate the thirst centers that are found in the hypothalamus, which will create a conscious then you're even going to feel nice. Okay, the water has been compensated. So now, having talked about the mechanism of thirsting, now we can also ask ourselves to say, what are the types of thirst that we have? So we have two types of thirsting. We have what we call hypo. Okay, so we had a network issue there. We are back. So we have two types of thirst as we are talking about. So we have what we call hypovolemic thirsty, as well as osmotic thirsty. Okay, so these are the two types of thirst that we have. So let's start with osmotic thirsty. So basically, osmotic thirsty, it is when your body loses water but not the solids. So meaning that the body will lose water but not the salt content of the body, or rather the solids. So when the body loses water, the blood plasma is going to be concentrated. That means that the osmolality is going to be increased, because the body solids are just being maintained, but the water is being lost. So that actually even reminds us of the term osmotic thirsty. It is just dealing with the loss of water, osmo. So the loss of water can be triggered by what you talked about, can be due to sweating, just not due to not drinking a lot of water. Let's say you are fasting and you say it is a total one, you are not going to drink water, and so on and so forth. You can have what we call osmotic thirsty. Then you can also have osmotic thirst, so it can be triggered or it can be caused due to eating too much salty food. So there are those snacks that are concentrated with sodium chloride, which is salt, and also just those people just like eating food that have got, high concentration of salt. You find that at the end of eating, you even feel thirsty, you want to go and drink water. So even sweating, even urinating can also be one of the causes of osmotic thirst, like frequent urinating, because when you urinate, you are losing also water. Yeah, so basically that is just what the concept that you need to grasp under osmotic thirst, that this is when the body loses water but not salt or solute material, rather substances. Yes, of course. Also, the other type of thirst, which is hypovolemic thirsty. So hypovolemic thirsty, just to break it down a bit, hypo means low, but this low, it is not the normal low, but it is lower than normal, which you can basically just say deficiency. That is hypo. Then volemic is just coming from the word volume. Yeah, so you find that hypovolemic thirsty occurs, or rather it is the type of thirsty that is due to the decrease in blood volume. So it occurs when you lose both water and solutes. So this type of thirsty can be triggered due to hemorrhage. So hemorrhage, this is basically just the loss of blood from the circulatory system. So it can be due to the rupture of blood. You are having an internal bleeding or an external bleeding. That's why we have external hemorrhage and internal hemorrhage. Yeah. So also vomiting. Also vomiting is one of the factors that can bring about hypovolemic thirst, because when you vomit, you are using together those electrolytes that are in this type in this type are being categorized as solutes, as well as water. Even diarrhea can be a cause. That's why you find that in those people who are suffering from diarrhea as well as cholera, they suffer from what we call hypovolemic thirst, because they are losing water, and not only water but also the solutes that are in the body, which in this case we can call the electrolytes, which are very, very important. So now there's a mechanism that we also have to understand under hypovolemic thirst. Like when you're having hypovolemic thirst, how does the body come in so that it can try to maintain its internal environment so that it does not disturb the normal functioning of the cells? So basically what happens is that when blood volume tends to decrease, because one thing that you have to know is that when blood volume decreases, also the blood pressure decreases. These two things, they work together. So what happens is that decrease in blood volume also causes decrease in blood pressure. So that decrease in blood pressure is going to be detected by what we call the baroreceptors. So these baroreceptors are just specialized in detecting pressure. So when you hear baroreceptors, they're just specialized in detecting the pressure. How they detect this pressure, it is in the stretching of the blood vessels. So we are going to find that you have baroreceptors in the aortic arc, which is the arc of the aorta. Then you also have baroreceptors in the carotid artery. As you'll be doing your anatomy, you know the carotid artery and the location where it is found, very, very important. So you're going to find that these are some of the blood vessels that has, even the heart has got these baroreceptors. So what happens is that decrease in blood pressure is going to be detected by the baroreceptors, because when the pressure is normal, there is just going to be a minimal and a required stretching of these blood vessels. So even the electrical impulses or the signals that these baroreceptors are going to be sending to the brain is just going to be normal. The body will consider it to be normal because the pressure of the body is normal. But in a condition where we are having decrease in blood pressure, going to find that these baroreceptors are not going to be firing the action potentials, or rather they are not going to be sending the stimuli, the signals, as frequent as they should be sending when they are normal. So meaning that signal will be detected by the hypothalamus to say the signal has reduced, that is coming from the baroreceptor, meaning that we have got decrease in blood pressure. So what the hypothalamus does is that it is going to cause the activation of what we call renin angiotensin system. It is going to cause the activation of renin angiotensin system. Yeah, this one you can also find it, they will say it as the renin angiotensin aldosterone system. So this system actually involves the formation of what we call angiotensin 2. This angiotensin 2 is actually a vasoconstrictor. It is a vasoconstrictor. So what happens is that when this angiotensin 2 is formed, it is going to cause vasoconstriction of the blood vessels so that it can compensate the blood pressure. We know that when you're having, let's say for example, you're having a pipe, what happens is that if the pressure of the water that is coming out of that pipe is low, how you can increase the pressure, it is by narrowing the lumen of the pipe. So you are constricting that pipe so that you can increase the pressure and the flow of the water out. And if you increase the lumen, you're going to find that the pressure will even decrease the more. So what angiotensin does is that it is going to go and vasoconstrict the blood vessels, and as it does that, it is going to increase the pressure, the blood pressure, and the rate of the flow of blood. And apart from that, it also goes to the centers that are found in the hypothalamus, those thirst centers, and it will stimulate them, and they will bring about a conscious awareness that you are thirsty and you drink. So how this angiotensin is formed is that the decrease in blood pressure is going to be detected by what we call the juxtaglomerular cells that are found in the kidney. So when these cells detect the decrease in pressure, they are going to release an enzyme that is known as renin. So this enzyme, when it is released, it is going to go and act on a molecule that is known as angiotensinogen. This angiotensinogen is a molecule that is formed or that is produced and secreted by the liver. So when it is when this angiotensinogen is released by the liver, the renin goes and acts on it. So it will go and convert this angiotensinogen into angiotensin 1. When this angiotensin 1 is formed, it is going to be transported and taken to the lungs, where in the lungs we are going to find an enzyme that is known as angiotensin converting enzyme. So this angiotensin converting enzyme will act on angiotensin 1 and convert it to angiotensin 2. And the same angiotensin 2 is what we know the vasoconstrictor, which will start now constricting the blood vessels to compensate the blood pressure. And it is also going to activate the thirst centers in the hypothalamus in the brain to bring about the conscious awareness that you are thirsty and you drink water. And it also goes to the so it also goes to stimulate the release of aldosterone that brings about the reabsorption of sodium. And we know that when there is reabsorption of sodium, there is also going to be retention of water, because sodium will create that osmotic gradient that will bring about the water movement towards sodium. Yeah. So having known this, these are the concepts that you have to understand about thirsty. So having known this, let's also talk about the cholera toxin and its mechanism. So how does this cholera toxin cause cholera, and what is cholera, and why is it that in those patients who are suffering from cholera, they tend to have a watery stool like you get? And why is it that these people, they tend to die within a few hours of getting infected? Because we know that the incubation period of... Okay, so I can see a question there from Leonard Musonda. Okay, so osmolarity. What we said under osmolarity is that I believe you are getting me. Okay, so what we said under osmolarity is that osmolarity, this is just basically the concentration of solutes, or the concentration of solute particles per kg of a solvent. And we said that osmolarity, it is measured in osmos per kg. So what the difference with osmolarity, the one for R, because we have osmolarity, just like the one that you have asked, as well as osmolarity. Yeah, so this one which you have asked, which is osmolarity, it is basically the concentration of solute particles per kg of a solvent, and it is measured in milliosmoles per kg, because we are measuring the concentration of those particles in 1 kg of that solution or solvent. Then the difference with osmolarity is that also osmolarity concentrates on describing or measuring the concentration of particles per liter, or the concentration of particles, but for it is per liter and not per kg. That's the difference. And its units, instead of osmos per kg, for osmolarity it is osmos or milliosmos per liter. That's the difference. And osmolarity. And we said that the preferred one in physiology, they like osmolarity. That is because osmolarity, it is more accurate and it is temperature independent, meaning that it is not affected by temperature. And it here you are free to ask the question. Okay, so let's dive into cholera toxin, the mechanism of the cholera toxin and severe dehydration. So cholera toxin, or let me just start with cholera. So cholera, we know that it's just a severe disease that is described by severe dehydration, where a patient tends to dehydrate severely, such that if there is no intervention, the patient might die just within the first 24 hours of getting sick. So we have got what we call cholera toxin. So this cholera toxin is produced by an agent that is known as Vibrio cholerae. Vibrio cholerae. So this Vibrio cholerae, we know it is a bacteria that is a gram-negative bacteria. So this bacteria has got a high resistance to antibiotics compared to these other bacteria that are gram-positive. Yeah, so for it does not really react the same way with other gram-positive bacteria because of how the nature of this bacteria. So this Vibrio cholerae, it produces what we call a cholera toxin. This cholera toxin, it has got two subunits. So it has got the A subunit, it also has the B subunits. That's the composition of this cholera toxin. So we know that the cholera toxin disrupts the normal ion transport in the intestinal epithelial cells. It will disrupt that. So you're going to find that it will disrupt the absorption of these ions, ions like sodium, chloride. It will disrupt that, and as a result it will cause severe loss of water, which is going to result into dehydration. So what happens is that when one is exposed to this causative agent, which is the Vibrio cholerae, this Vibrio cholerae basically is going to secrete a toxin. So let's use this table so that we understand nicely what I'm talking about. So what happens is this is the lumen of the intestine. So we are talking about an intestine. This is an intestine. This is the lumen where we are going to see the movement of the food materials that are going to be passing here. Those electrolytes will be passing here. Those are about to be absorbed. So what happens is that when one is exposed to this bacteria, which is Vibrio cholerae, it is going to release a toxin that is known as cholera toxin, of which this toxin is made up of two subunits. We have got A subunit as well as the B subunit. So what happens is that when this cholera toxin, which is this point here, this toxin is released by bacteria, it is going to go and bind onto the enterocyte receptors. So these enterocyte receptors are receptors that are found onto the enterocyte. These enterocytes are cells of the intestines. So when it goes and binds to these receptors are known as the GM1 ganglioside receptors. GM1 ganglioside receptors. When it goes and binds there, what facilitates the binding of this toxin is the B subunit. When the B subunit binds there, the A subunit is going to be endocytosed into the cell. So before we go to the pathway of where the A subunit is going to go, let's understand how the normal pathway works. So we are going to find that if there's a stimuli onto this receptor, which is the GM1 ganglioside receptor, we are going to find that we are going to have what we call a G protein coupled receptor signal transduction. So this signal transduction, we are going to find that if a stimuli binds to this receptor, it is going to cause the dissociation of this molecule, which is here. So this molecule is a molecule that will be having an alpha subunit, or rather the gamma subunit, the beta subunit, and alpha subunit, which are attached to a molecule which is the GDP molecule, which is the guanine diphosphate molecule. So when this happens, this molecule is going to dissociate, and as it dissociates, it attaches away from the receptor. This molecule is going to be activated from GDP to GTP. So meaning that there's going to be a usage of an ATP molecule, whereby this guanine diphosphate is going to be converted to guanine triphosphate, meaning that the phosphate molecules are going to be changed from 2, that's why we are here in diphosphate, to 3, which is triphosphate. So when that happens, this alpha subunit together with the GTP molecule are going to be detached away from the gamma and the beta subunits. When this happens, this is going to be in its active state, in which it will be having the GTP molecule here. Then this subunit will move and go to an enzyme. So this enzyme that we are seeing here, it is known as adenylate cyclase enzyme. So this adenylate cyclase enzyme, it has got a binding site for this O molecule. So when this molecule goes and binds to this adenylate cyclase enzyme, this molecule will cause the activation of these adenoside cyclase enzyme, adenylate cyclase enzyme. So when this enzyme is activated, what it will start causing is that it will start converting ATP into cyclic AMP. So cyclic AMP is basically cyclic adenosine monophosphate. So the ATP will be converted from adenosine triphosphate to cyclic adenosine monophosphate. So when these cyclic AMP molecules are formed, they are going to go and activate an enzyme that is known as protein kinase A enzyme. When they activate this protein kinase A enzyme, this protein kinase A enzyme, then it is going to go and do the function that it is required to do, depending on what was the stimuli there. So now, in a condition whereby a toxin is released and binds to this receptor, which is the GM1 ganglioside receptor, the B subunit will facilitate the binding, and the A subunit is going to be endocytosed into the cell. When it is endocytosed into the cell, it is going to be transported to the endoplasmic reticulum. There in the endoplasmic reticulum, the A subunit is going to be activated, and as it is activated, it is going to cause also the activation of an enzyme that is known as ADP riboxyltransferase enzyme. This ADP riboxyltransferase enzyme, it goes to this molecule here, this molecule having the alpha subunit and the GTP. What happens is that this ADP riboxyltransferase enzyme will cause ADP riboxylation, of which this riboxylation is basically just the change in the conformation of this molecule, such that this molecule be locked in its active state. So meaning that this molecule is going to be activated, and it is just going to be activated just like that. And when it is activated continuously, it is also going to cause continuous activation of an enzyme, which is the adenylate cyclase enzyme. When this enzyme is also activated, it will also cause the conversion of ATP to cyclic AMP. When that cyclic AMP is produced due to the continuous activation of this enzyme, they are going to have built up of this cyclic AMP. When this cyclic AMP builds up, it is going to activate a protein kinase A enzyme. So what happens is that this protein kinase A enzyme, when it is formed, it is going to go and bind to channels that are found in on the basal apical membrane, or the apical pore of these cells. So when it goes to these channels, these channels are known as the cystic, just a minute. So these channels are known as cystic fibrosis transmembrane regulator chloride channels. Cystic fibrosis transmembrane regulator channels. Yeah, in some other books you also find it being written as cystic fibrosis transmembrane conductance regulator chloride channel. So this channel basically is just a channel that facilitates the entry, or it just facilitates the movement of chloride ions. So what happens is that these chloride ions, when this channel opens, it is going to start allowing the flow of chloride ions into the lumen of this intestine. So it is going to allow the flow of this chloride ion. So this chloride ion will start moving from inside of the cell getting into the lumen, because as long as this molecule here, it is continuously activating also this enzyme also activating, causing the conversion of ATP to cyclic AMP, and this also continuously activating. This means that the protein kinase A will also cause continuous opening, or rather continuous activation of this channel, causing them to open and allow the flow of chloride from inside of the cell to the outside. So as this happens, the chloride ions will start moving into the lumen. And also what happens is that the same protein kinase A enzyme, it goes and affects what we call the sodium hydrogen exchanger. So what happens is that this sodium hydrogen exchanger, what it does is that it exchanges sodium molecule. Normally the sodium molecule will be taken inside, and hydrogen ion will be taken outside. So it will be exchanging a sodium ion for an hydrogen ion. So that's how normally it works. So in the presence of continuous activation of protein kinase A, you are going to find that this enzyme is going, or rather this channel is going to be disabled. It will stop working. As it is disabled, it means that there won't be any absorption of sodium. So the sodium will start now building up into the lumen. And we know that when sodium and chloride builds up into the lumen, they are going to increase the osmolality of the lumen. And the osmolality of the lumen starts now increasing. It will start allowing now the flow of water from inside of the cell getting into the lumen. And as water gets now into the lumen there, the person will start now dehydrate. They will start undergoing severe dehydration. So this is why even in patients with cholera, who are suffering from cholera, they tend to have a watery stool, meaning the stool that is too concentrated with water. That is because this whole mechanism is what happens. So in as much as they are also losing water, there is also another consequence of them losing electrolytes. That's why they tend to die within just the first 24 hours. Because there is loss of electrolytes, because these electrolytes are very, very important in the functioning of the vital organs and also in the functioning of the body systems that we are going to learn more about. So basically this is just the whole mechanism of how the cholera toxin causes cholera and also severe dehydration, the whole condition. So if you have questions, feel free to ask your questions. If you are still not clear, still comment and ask your question. Not until you are clear, that's when you are going to be happy here, because we are here to help each other. Otherwise, for those who have persevered up to the end of this lecture, thank you very much. So if you have any questions, feel free to inbox or to just send in the group. Then we'll be happy to attend to your questions. This marks the end of our lecture under physiology of thirsty, as well as the mechanism of cholera toxin and also severe dehydration. Otherwise, thank you very much. Have a blessed night.